Hi, I'm Paul Macklin. I'm really happy and excited to welcome you to the 2023 PhysiCell Virtual Workshop and Hackathon. This is session 0. We're going to start introducing the basics of agent-based modeling in PhysiCell, and this will be the launch point for this year's training materials. So the course goals for this year's virtual workshop and hackathon is first to learn about agent-based modeling in biology, to learn about key cell interactions in multicellular systems biology in particular, and how to model those in the PhysiCell simulation platform, and how to build and explore these models together. If you are a participant in the hackathon, we will, in addition, form new teams and build either a PhysiCell model of a specific multicellular system, such as in cancer, or to build a PhysiCell tool or extension that helps out the community. We're also excited as we welcome you to this workshop and hackathon to start teaching the next generation of leaders in the PhysiCell community and to hopefully provide a great starting point for you to run your own workshops and hackathons in your own local communities. In this particular session, we're going to be learning about the motivation for and concepts of agent-based modeling. We'll briefly survey some of the main types of agent-based modeling approaches and contextualize PhysiCell as an agent-based modeling approach within the broader context of these types of approaches. And we'll close out with some examples of building models in PhysiCell that might help motivate some of our work through the coming sessions. So first of all, one thing that interests a lot of us as scientists and biologists is to understand cancer ecosystems and other multicellular ecosystems, understanding really how cells, when you get down to it, have a handful of individual behaviors. They can grow, they can divide, they can differentiate or change type, they can die, they can consume things in their environment or sample them, they can secrete things, metabolize them, they can secrete signaling factors and things like this. Of course, they can mutate to change types, and they can move around. So these are some of the early single cell behaviors that they have. Then there are a handful of cell-cell pairwise interactions, like they can stick to each other, they can push each other around, they can consume or predate or eat each other. And of course, they can do other things through their contact interactions. They can hunt or attack one another, for example. And then with these constraints and these interactions, they act in an environment with physical constraints. They must receive substrates like oxygen and glucose through diffusion. And there are diffusion limits to cell-cell communication by chemical diffusion. And of course, they run up against mechanical barriers and other constraints in their environment. And somehow, the combination of these single cell behaviors, pairwise interactions, and physical constraints, when you scale it up over tens of thousands or more cells interacting in 3D environments, lead to the complex ecosystems you see in systems like cancer and other multicellular systems biology. And so multicellular systems biology is seeking to understand these systems, how individual behaviors, simple cell-cell interactions, and physical constraints combine to lead to the emergent behaviors we see in biology and in tissues and other ecosystems. And then conversely, we're not interested in just understanding the systems, but trying to control them or re-engineering them. And so when we've moved from multicellular systems biology of understanding the systems to actually applying that knowledge to control them, now we've moved into the realm of multicellular systems engineering. And so that's really the goal of our field as we move forward. And so I like to think of multicellular systems biology as a play, that the microenvironment is the stage and the cells are the actors upon that stage, and they follow their own scripts as they move around through their environment and interact. But just like a play, the scripts can change based upon the scene or the stage. So this is the fact that the cells have a microenvironment dependent phenotype, the way they behave. Likewise, as in a play, the cells aren't just all talking out into the void, but they're talking to each other. So the dialogue is critical. So just like in a play, cell-cell communication is critical to understanding this play that we see. The actors can rearrange the stage. So let's kind of think of it as avant-garde modern theater here, but the cells can rip up and tear up and remodel and rebuild the stage as they're in the middle of the play. And of course, particularly in diseases like cancer, sometimes the actors ignore the scripts altogether and ad-lib. And this would be the case of mutations and other adaptations that we see in cancer biology and other fields. And so it's our job as scientists to figure out each actor's script, that is, the rules of each cell and their individual scripts by watching this play over and over and over again. You take a handful of cells or actors, place them in a scene, press play, hit action, watch the play, make your observation, say, okay, I think they're interacting this way. And we start to kind of hypothesize what the rules are of this play. And if we can change the scene, change the players, change the conditions enough times, we hope that we get a more and more complete understanding of what's going on in this play. So we want to watch the play many times, make hypotheses,
and test out those hypotheses to see if we can recreate the play and understand the system. And then as engineers and clinicians, we want to not only understand the play, but we want to rewrite the script so that the players will behave and form well-functioning tissues. So agent-based modeling is a modeling paradigm for these kinds of complex systems. The idea is that cells are software agents, or little individual pieces of software with well-defined attributes and processes that can move around and interact in virtual tissue environments. So that's really what we're going to be covering in this course as we go through. So to understand an agent-based model, we need to at least have a notion of what we mean by a discrete model. So discrete here doesn't mean that we're being, discrete and subtle. It means discrete mathematics. And so one way to describe discrete models is actually to describe something that might be more familiar, continuum mathematics. And so a continuum model in mathematics means a variable that can take on continuous values. It can go, say, between negative infinity and positive infinity, anywhere between 0 and infinity, and any number in between, like fractional numbers and irrational values. And typically, we describe these kinds of continuous variables with a differential equation that can have nice, smooth derivatives and things like this. So the classical example that you might be used to is exponential growth. So the continuum variable here might be cell density, rho, which we might model as population growth, saying that d rho dt, the change rate of the population, is proportional to the size of the population. So if you have more cells, those cells can all divide. So the greater the cell population, the greater the overall population growth rate. So d rho dt is proportional to rho. So that would be an example of a continuum model. Discrete models are different, where we are tracking distinct individuals with discrete events. There can be division events, there can be death events, there can be movement events. And so the variables tend to take discrete values like Boolean, true or false, or integer, 0, 1, 2, 3, or 4 cells, but not 4.13576 cells. And so an example of a discrete model would be a classical birth-death process in stochastic processes. A cell population x of t, well, x is the number of cells in our population, so it can have an integer value. And it models these as birth events with a birth rate r. Between now, time t, and a time a little bit further from now, some discrete chunk of time, t plus delta t, you might have some probability, in this case, the birth rate times delta t, that there is going to be a birth event that increases your population by 1. And so you can actually write this as a finite equation. It's just an equation saying that the probability of that the population at the next time is n plus 1, given that right now it's n, is this r times delta t. And then you start adding these up and get all sorts of things that start looking like a discrete model. An agent-based model is a particular form of a discrete model with a little bit more detail, that we model the cells here as individual models, as individual entities that move around in our environment. And so we have a finite number, so there's a discrete part of cell agents, but then we start layering on the individual biology of these agents. And so we'll often combine the agent-based model with other models of our biological system. For example, we might model cells as discrete individuals with a specific location and size and type. And on top of that, say that they live in an environment with an oxygen distribution and use a diffusion equation to model the movement of oxygen. And so we'll put those together, saying that this hybrid of a discrete model of the cells and a continuum model of the environment, put them together, and this is something that is often called a hybrid discrete continuum model. Object-oriented programming is something you'll see oftentimes in agent-based modeling. And the idea is that really this is actually an ideal case for object-oriented programming. Here, the objects in our software environment of our simulation are individual cells. So each one of those cells is an object, a member of some kind of cell class. And as a member of that class, each cell already knows something about the key biological processes that we've already modeled for cells in general, and some individual parameter values that describe the state of that individual cell. And we can update all those things over time. And these agent-based models can be made to be a little bit closer to the biology. We look through our microscope together and we see individual cells. And so we kind of copy over those properties from what we see in a real microscope into our virtual microscope. The cells have position, size, type, a cell cycle status, maybe a likelihood of dying, some mechanical properties like a Young's modulus, for example. And you're going to add that detail onto your individual cell objects in your environment and then define the rules of how those objects interact.
And so you can tailor the level of detail of an agent-based model to the types of data that you expect, the types of observations that you see, and the types of things that you might want to predict. And in some models, you might go down and add a whole lot of detail, even down to molecular level detail. Things like receptor trafficking models might be added into an individual model, or models of protein interaction networks and protein signaling networks. So it really depends upon what you're trying to do. In a typical agent-based model, they all have a fairly similar flow. There'll be setup steps and then main loop steps. So setup will be things like reading parameters to say, how do I want to set up the simulation? Then set up your microenvironment, particular diffusible things like oxygen, glucose, or drugs, and create meshes and set up all the solvers for those problems. And then you want to set up your cell agents. Say, what are the cell types in my particular simulation? Do I have cancer cells and T cells and macrophages? Well, then let's define all those types with their own individual parameter values, and then we'll instantiate the cells, which means we'll place them in our environment. Okay, well, now we're set up. And now it's time to do the main simulation loop. Now, recognizing that in biology, most things are connected, this means we have to take time into little tiny slices and alternate all of our processes. So we'll say, hold all of our cells fixed for a moment, and we're going to update environment. We'll solve the diffusion equations. We'll solve secretion and uptake and consumption of chemical factors. So say, okay, all my cells, some of them are secreting things, some of them are absorbing it. Now I'm going to take those new things in the environment and diffuse them around to kind of transport it. Hold that fixed now, and then say, now let's update all of our cells based upon where they're in the environment, sample the environment, based upon your hypotheses, update their process properties, like how fast do they want to cycle, how fast do they want to move, how likely are they to do a cell cycle transition or change to a new type or differentiate. And then update all those processes that are attached to the cells. So go through all the cells, update their properties, run their state models like through cell cycle progression and all of that. And then you can say, now let's do the mechanics. Let's calculate the forces exchanged between the cells, and then update their velocities, and then update their positions. Say, okay, now I've done it. I've update time a little bit more and move on. And we just continue. So we say, freeze all the cells, up to the environment, freeze the environment, update all the cells, and just keep alternating this as we march forward in time and finish our simulation. There are many ways to actually do this, though, and it really depends upon how you want to model the environment and the cells. And so the main two classifications that we like to think of on agent-based models are lattice or off-lattice models. So in a lattice-based model, let's kind of this column on the left here, you divide space into a bunch of lattice points. And each lattice point can hold, let's say in the simplest form, each lattice site can hold exactly one cell. That would be the case of, say, a cellular automaton model. So if you want to model a cell, you basically say, where am I in this lattice at some I, J, K location? And should I move left, right, up, down, diagonally, vertically? Well, those movement rules are going to happen in discrete hops along lattice sites. And so those models can be very simple to write and to code and to implement. They can be very, very computationally fast, but they can have their own issues, in particular that the accuracy of treating all cells as identically sized and holding identical positions can be really problematic. It's very tough to do mechanics in that kind of a model. So one solution to that is to actually divide them up into smaller lattices, say that every cell might actually be several lattice sites. That's the typical way you do it in a cellular POTS model, such as PhysiCell 3D. And that can be a really great way to get cell morphology or shape, but it's very, very computationally expensive. And, it has some difficulty sometimes in relating the thermodynamic steps in those codes to the physical steps that we're used to in biology. So all methods have pros and cons. And so lattice-based models have a lot of great uses, but have some key drawbacks that we're concerned about. On the other side of the equation, there are off-lattice models. This is where a cell can have any position in space. It doesn't have to be at a specific lattice site. It just has X, Y, Z coordinates. And then the way you update the position of that cell is by looking at who is nearby and doing some kind of a force interaction calculation. Say I'm near these three cells, boop, 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 and I have an adhesive force to them, and they have a repulsive force pushing me away to resist overlap. I might have a little bit of a locomotive force this way. I add up the contribution from all these forces, and that tells me my acceleration and my velocity to say my cell's moving this way. And so you think of it as like a massive n-body problem from physics, and you keep updating the positions. Now, like any other n-body problem, the complexity of this center-based approach can grow very fast. That if I have n cells,
that must interact with n-1 other cells, that means the computational complexity increases as a square of the number of cells. That's a real problem. And so we have a lot of tricks to bring that back down to order n, but it takes some work. On the flip side, you can have all the cells can have completely independent sizes and positions that are not bound to a lattice. And so that's the approach that we take with some pros, that it's still fairly computationally cheap and that we can scale it up. Some cons, that is not as fast as the cellular cellular automaton model. And it doesn't get, say, the shape of a cell like cellular pots. So there, like all methods, we have some trade-offs. If you want to look at a deeper review of types of agent-based models, we have a review article we wrote in JCO Clinical Cancer Informatics several years ago. If you download the slide deck, you can find the PDF link to that paper, and I really encourage you to give it a look. So where does PhysiCell fit into this? Well, we're an off-lattice model. Our cells can have any position, not bound to lattice. They can have any size, not bound to those grid sizes. And so that's the important thing. The spatial resolution of PhysiCell is that each agent right now represents one cell, although there are some tricks to either represent clusters of cells as agents or smaller pieces of cells as agents, but that takes a little bit of extra work. PhysiCell couples this discrete coverage of individual cells with microenvironment modeled as partial differential equations to model transport of chemical substrates, and that makes it a hybrid discrete continuum approach. And I want to note that PhysiCell, thanks to our amazing community of collaborators, can hook into other modeling frameworks, combine them to truly multicellular, multi-scale frameworks. In particular, we connect with PhysiBoss to model Boolean networks for cell decisions. So you'll hear about that later in this session. You can also connect PhysiCell to ODE models of subcellular dynamics and molecular dynamics. And there are other approaches too to get different scales of interaction into PhysiCell by connecting to other pieces. And so we'll learn about that more this week. So to kind of explain the key pieces in PhysiCell now, one of the most important things of PhysiCell is BioFBM, a finite volume method that's geared towards these kinds of biological problems. The idea that's that we want to model many cells as discrete sources and consumers of diffusing chemical substrates. So and we knew that we had to do more than, say, 1 or 2 diffusion equations, but vectors of diffusion equations, 5, 10, 15 diffusing factors in 2D or 3D. And so we took that and we built it as a highly optimized technique with some parallelization. It works on Windows, it works on Linux, it works on Mac OS, it works on other platforms with no changes to the code. And this is kind of like the initial foundation of PhysiCell, saying the cells are in fixed positions, just like at the beginning of our update space. Update states that we talked about earlier on in the general technique or flow. And then those cells can secrete things, consume things, and then we model the diffusion of those 15 or 20 diffusing substrates. And so that's all open source and published several years ago in bioinformatics. So then we took that framework and said, now that we have a bunch of cell agents that can secrete and consume things, let's start layering in the biology. And that's where PhysiCell was born. Is that each individual cell agent is now coupled to that diffusion environment that we just talked about, but on top of that has mechanics-based movement, built-in models for cell cycling, death, and other key biological processes, realizing that we're going to need these models all the time. And so we built them in. They're all built in. You don't have to write your own code to say, how do I take a cell, divide it, and place its daughter cells, and update data structures? That's all automated in PhysiCell. But it's very customizable. You can add custom functions on a cell-by-cell basis that says, how do I change my behavior as a function of the things around me? And so you can write those functions to codify your biological hypotheses, and that becomes your model. So we provide this general framework on top of which you can add your hypotheses. But we take care of all the low-level stuff like the mechanics, movement, cycling, and you take care of saying, how often do the cells cycle? How much do they move? When do they want to do different things? So you can kind of focus on the biology and not the low-level coding. Now, all this is also cross-platform. The same code runs on via C++, on Windows, on Linux, on Mac OS, and other platforms. You can take a code that you developed on your desktop, and then when you're ready, go out and run it on a cluster and do 100,000 simulations and start doing something with it. And that's kind of the idea here. So here's a typical 3D example, showing also the heterogeneity that you can model with an agent-based model. Every one of these cells has a different likelihood of dividing, shaded as a color from blue, saying not very dividing, to yellow, very aggressively dividing. On top of that, every cell requires oxygen to divide. So the more oxygen the cell has, the more likely it's divided.
going to divide. And the less oxygen, the less likely it is to divide. And if oxygen goes too low, then the cell would become necrotic and start dying from lack of resources. And so in this simulation, on the outer edge, oxygen is set at a Dirichlet condition or a fixed condition, diffuses into the environment. The cancer cells consume that oxygen, so the oxygen levels drop as you go into the tumor until you get too far into this thing, and you'll get these brown necrotic cells that emerge. So each one of these dots is a simulated cell. And then here we have just a cross section so you can see the interior structure. So watch the individual cells that move around. Yellow cells are giving birth to yellow cells, blue cells to blue cells. You can see that these foci of highly proliferative cells start to emerge, which makes sense. They divide faster. And then when you have that two conditions combined, that the fast dividing cells reach the outer edge of the tumor where they have more access to environment, they just completely take off and completely outcompete everybody else. The other thing that you see emerging as emergent dynamics here, if you look into the core of this tumor, you can see the brown necrotic region. So oxygen has dropped as it gotten into the interior. And then you have this brown region of necrotic material. And here the mechanics are a bit interesting, right? Because you have cells are still sticking to each other, but shrinking, and that's mechanically unstable. And you get the emergence of those chasms of fluid-filled cavity emerging in the necrotic core, which is something we actually see in hanging drop spheroid experiments. The other thing that's interesting is to watch the flux of individual cells. On the outer edge, when cells proliferate and divide into two daughter cells, it's mechanically easier to just push your daughter cells towards the outer edge of the tumor, and this whole thing starts to grow. But now look at the, I'm going to go ahead and start playing the movie again, but watch some of these cells near the interior. These cells might divide, and mechanically, is it more favorable to push against 15 or 20 cells this way, or against a few cells into this empty space? Turns out it's easier to push into the necrotic core. So you'll get some flux of the cells going outward and some flux of the cells being pushed inward. And so that's something that's an emergent dynamic of all the rules we have in our simple hypotheses we have in our models. See how this inward flux starts pushing cells into the edge of the necrotic core. So there's an example of a model that we can build in PhysiCell. So key parts of a PhysiCell model include, first of all, the stage or the environment where we define diffusible substrates. And so for each of the substrates, you need to define a diffusion coefficient and a decay rate, and then how you treat the boundary of this. And that's defined in a markup language that is now editable in a graphical studio that you'll see in future talks. After you define the stage, you need to define the types of players in your model. So we're going to come up with cell definitions. Say, here is the definition of cancer cell, of a motile cancer cell, of a fibroblast, of a CD8 positive T cell. So each cell type will have a name, a default phenotype, and then we'll define that in our configuration file, which we can all do by a graphical interface. Once you've defined the stage and the types of players, then you're to place the individual players in your simulations, say which cell types are there. So you kind of clone one of the cell definitions and initialize it based upon that definition. Then every cell will have its own state variables, things like its position, mechanical pressure, interaction lists, things like this. And then the phenotype, which defines the bundling of the behavioral parameters, and then functions that act upon that phenotype to change it dynamically in time. And so really our entire workflow is centered around modeling the cell's behavioral phenotype. We have the basic processes already built in: cycling, death, volume, regulation, mechanics, things like this. The things that you need to model as modelers to say, how often does a cell want to cycle? How often does a cell want to die? What rate is it sticking to other things and moving around? At what rate is it secreting things, consuming things, and how does it interact with other cells? So you really get to focus on the behavioral hypotheses, which you're going to write as functions and not the low-level stuff. New in PhysiCell this year, we're really excited to introduce actually in this workshop, is that we have a way to kind of simplify the way we build these models now. And so we have things called signals. These are the inputs to cell rules or hypotheses. These are things in the environment that can influence a cell behavior. And in particular, we auto-generate a dictionary with intuitive human language symbols like oxygen or glucose that you can query in your simulation models as inputs to your models without a very complicated syntax. Likewise, behaviors are the outputs of the cell rules, and those map onto the behavioral parameters in PhysiCell, things like migration speed, secretion rates, cycling rates. And we have an auto-generated dictionary of, again, intuitive human language symbols like cycle entry or apoptosis. And you have easy functions that can write those particular behaviors to control your cells and your models. And now the new thing here is that we can combine the signals and behaviors and grammatical rules of a behavioral grammar. say that, and these are human readable hypotheses, signal S, say oxygen, increases or decreases behavior B, say cycle entry. And we can write those rules in a graphical studio without writing a single line of C++. And then we will parse those rules at runtime to auto-generate the code. So now you can create a model, set it up, and write your hypothesis rules graphically and build a very substantial model without having to write a whole lot of code. And so that's our goal as we progress with PhysiCell. I want to just provide a quick note that PhysiCell does have a number of time steps to think about because biology has a number of time steps. And so our very fastest time step is diffusion. We right now default to 0.01 minute time steps to get accurate and stable diffusion solutions. Mechanics is the next fastest because cells, molecules move very fast. Cells move kind of fast in the order of microns per minute in typical mammalian biology. And so we have a 0.1 minute time step by default for mechanics. And then cells change behaviors a little bit slower than that. So we say, oh, that's happening on an order of hours to half hour. So we give a 6-minute time step for that. And this allows some significant efficiency improvements in the code. And then intracellular models can have their own step sizes. Okay, so we spent some time kind of describing what agent-based models are and some notion of it, but now let's try to kind of clean it up and make it even more concrete by going to some recent modeling examples. And so the first example I'd like to present is work by a former graduate student named Yafei Wang here at IU, looking at the interaction between the liver parenchyma, or normal tissue, and colorectal cancer micrometastases. And so, we had looked at a lot of the literature. This is joint with Shannon Mumenthaler in particular at USC. We had looked at a lot of literature, and some, the molecular stuff had been already quite investigated, and some of the small scale mechanics have been investigated. In particular, if the ECM has certain signaling molecules bound to it, how does that change the behavior of either liver cells or tumor cells? Or how do signaling factors affect the individual behaviors of the individual cells? But we want to ask more about what happens at the larger scale, the tissue scale. How does compression of the liver tissue affect the micrometastases? And how do the micrometastases, through the mechanical interactions, affect those tissues? And so we built a basic model that found a way to say that if a cell is compressed by high pressure, that would downregulate cycle entry, because that's kind of a feedback you would see in actual biology. And on the flip side, the cells were imparting an elastic force on the surrounding tissue. So you push the tissue past its original configuration, it wants to elastically go back to its original configuration, and in the process, imparts an elastic force back onto the tumor cells. But if you keep a tissue deformed long enough, there could be a plastic reorganization where that becomes its new resting position. And the last thing we add is that a hypothesis that if you deform a tissue too far before it has an opportunity to kind of mechanically rearrange, that can be very stressful for normal tissue, and it can undergo a death under prolonged sustained deformation. So we took these very simple hypotheses and explored what could happen in a tumor. In particular, we took a very large section of liver and put a little tumor micrometastasis in the center, and we just plotted its growth under these typical rules here. Now, interestingly enough, yellow cells are the what we did is we added this pressure feedback, right, saying that higher pressure will downregulate cycling and eventually inhibit it. And so any cells undergoing very high pressure, causing it to arrest its cycle, are colored yellow. So the interesting thing, right, is as a tumor grows, at first that pressure is kind of distributed more towards the middle of the tumor. But then if a tumor gets big enough, that necrosis, right, that shrinkage of the necrotic material can kind of provide a pressure relief. And so in the inner part of the tumor, there's not so much pressure. So pressure is not arresting cycling, but the lack of oxygen is. So you can kind of get these different feedbacks until eventually you kind of get the structure, right, of an outer rim with high pressure, very outer rim where there's the proliferation into the invading tissue, and then an inner necrotic core. And if you take a cross section, you can see how even far away from the tumor, there is pressure and deformation that dissipates into the surrounding tissue and reaches a peak at the tumor interface, and then the pressure actually peaks a little bit behind that tumor edge, then decreases as you reach that necrotic center. And so that's something that was interesting. And then we started to ask, actually turned out that for some parameter combinations, this balance of the tumor growing in and imparting forces on the surrounding tissue imparting elastic forces back on the tumor, compressing it, that in some cases, these can come into balance. that in a sufficiently elastic tissue with a slow plastic restoration and a high tolerance to that deformation can actually completely arrest a tumor and keep it relatively dormant over very long periods of time. So that was actually quite interesting to find that pop out of this very simple model. And then the next question you say it can take is take a dormant micrometastasis and grow it for 90 days, and nothing happens. And so as our control, we're going to say nothing changes, just simulate another 90 days, and you see that metastasis stays pretty dormant for very long periods of time. But now we could try mechanical perturbations. Say, what happens if you reduce the elasticity of the surrounding liver tissue, and that perturbation can cause that formerly dormant tumor to start growing very quickly. Or you can reduce the tolerance of the surrounding tissue to deformation, and that perturbation can also lead to a runaway reawakening and runaway growth of the tumors. So in very simple model like this can predict already some interesting effects that mechanics can kind of be an uneasy balance between the tumor and the environment, and can keep something small for a long time, but some perturbations can reawaken an otherwise dormant tumor. As another example, we worked with Danielle Gilks and her collaborators at Johns Hopkins University looking at hypoxia-driven breast cancer invasion. And so Danielle's lab developed this amazing marker system where these breast cancer cells are initially red, but under sustained hypoxia or low oxygen conditions, they will eventually snip out the red fluorescent gene and permanently express a green gene. So if a cell has changed colors permanently to green, then you know it's either now hypoxic or at some point in its history has been hypoxic. So you can actually track its fate. And so this is really fortunate because we were able to ask new questions with this kind of system, saying, what are the rules of a hypoxic cancer cell? If you take a hypoxic cancer cell, or cancer cell put into hypoxic conditions, we know it's going to adapt. We know that it tends to downregulate adhesion, it tends to become more motile, and we know it tends to change its metabolism. So the bigger question we had to say, take this hypoxic adapted cell, put it back into normal conditions, dial back up the oxygen. What happens? Will this adapted cell keep its new phenotype forever, stay adapted, or will it go back to its old pre-hypoxic behavior? And if so, how long will it take? And if you query a room full of biologists, you can get answers that pretty much say that cell, over the whole range of possibilities, saying that maybe the cell will never reacquire its old behavior, maybe the cell instantly requires its whole behavior, or maybe it waits some time and then requires its old behavior. And probably, depending on the context, all these answers are right. So it's an interesting question. And now here's a neat imaging system that can start answering these questions, especially if we put it together with a computational model. And so we built a model. This is work led by Eva Rojo. We simulate oxygen diffusion in the environment, like our typical models. Proliferation and necrosis vary with oxygen and mechanical pressure, just like in some of the prior models we've shown you. Live cells could be normoxic or red, or hypoxic or formerly hypoxic green, and that when the cells are in low oxygen conditions, they will gradually change from red to green and change their behavior. And so we modeled that the green cells have increased migration and possibly changed proliferation and adhesion. And then we had a family of hypotheses, basically a sliding control knob that says, when those green cells reach normal oxygen conditions again, how long do they keep their old behavior before reverting back? Their adaptive behavior before reverting back. We call that the phenotypic persistence. How long does their new adaptive phenotype persist when they leave the bad conditions, the hypoxic conditions? And so we explored this broadly, this family of models. And so the first kind of our control, if you will, is assuming that there's no phenotypic persistence. That says that the moment that a hypoxic cell escapes its hypoxic conditions, it goes back to its old behavior. And so we got basically these very stable ring-structured tumors to grow, where the outer edge would be red because those cells had never been exposed to bad conditions. The inner band was green. These are cells that had been adapted to hypoxia and had migrated just long enough to escape it. And so they kind of reach the edge of the danger zone and then stop. So the tumor grows a little bit. Well, that means the oxygen gets a little bit more depleted. So those cells now migrate a little bit more, but they just kind of keep pace, and no cell ever escapes. Of course, if that worked, if that were true, if those hypotheses were right, then we would never have to worry about cancer invasion and metastasis, which unfortunately we know isn't true. So we would say that hypothesis of no phenotypic persistence is probably wrong. So the next thing we looked at was a permanent phenotypic persistence. So suppose now that when a hypoxic cell leaves the tumor, it keeps its new adaptive phenotype forever, which in our case we found was increased migration and slightly changed proliferation. Well, here you get cells that definitely make it out of the tumor and stream and move along, but they never slow down and colonize anywhere. So that may or may not be true, but that at least is more true than the no persistence phenotype because they broke out and broke free. Then the interesting thing we found was an intermediate hypothesis of a transient phenotypic persistence, saying that they leave the tumor and due to some lag in the system, for example, however long it takes to switch genes and change behaviors, the cells retain their migratory phenotype for a little bit of time after getting out of the bad conditions. And that matched our hypotheses really well in our observations. In fact, it predicted this really neat feature of these growing green hypoxic plumes that once one tumor cell would find a mechanical weakness, this green cell, and invade into the normoxic tissue, it kind of drove a little wedge and opened up a little opening for other migratory cells to exploit. And so you get this growing plume of green cells into the red material. We were a little worried about that, but then we talked with Danielle, and she showed us her mouse model and said that she had seen the exact same structures in her mouse structure. So this is actually very interesting. We took very simple hypotheses of cell behavior from Daniel Gilks's lab. And then we worked behind the scenes to try to calibrate migration of the green and red cells separately. And then we just moved and varied the knob of how long the cells had their persistent phenotype. And for intermediate values, we got structures that emerged completely out of the model that predicted what we saw in the mouse model without us ever knowing this. So just like when you build a machine learning model, you want to have training data and validation data. Well, in agent-based modeling, you want to have training hypotheses and validation hypotheses. The hypotheses that you use to build your model, and then hypotheses that you don't use to build your model, and you hope that emerge in your simulation as validating hypotheses, as validating observations. So the generated hypothesis by this model was that we would form these green plumes. Here you can see them in the dynamic model. And that was validated because we actually found those structures in the mouse model without it being part of what we used to build our model. And one thing we can point out here is that those same observations, we built them in a 2D model for simplicity to keep it fast, but then we verified that the same observations happen in 3D. So that was something that was quite fun. So one last example I'd like to give is some COVID modeling driven by a postdoc named Michael Getz in our lab and by a large coalition of over 40 contributors from 20 institutions across the US and internationally. So we're really grateful for this group. And so basically we were building iteratively a model of COVID SARS-CoV-2 virus landing in the lung tissue, infecting individual cells. There would be a replication model in each individual cell. And then the virus would be released and start infecting neighboring cells. And then on top of that, we added an immune model. And so we built this thing iteratively. Our very first prototype, we actually built overnight using an earlier nanotherapy model, where we had a virus landing on the tissue, a really basic ODE-based model of viral replication dynamics, and then viral release and diffusion in the environment, and a cell death response. And that we kind of used as our starting point and started recruiting team members. In the second version of the model, where we're continuing to train team members how to use the code, we added a basic receptor dynamics, saying that a virus in the environment had to bind to a receptor and come into the cell as an entry point. And then we added random seeding of the virus across the tissue. The third version of the model is where things got really interesting, that Adrienne Jenner and Amber Smith and a whole lot of Penny, for example, a lot of our collaborators came up with this wonderful agent-based model of immune cell behaviors now. So they started adding models of macrophage behaviors, T cell attack, and neutrophils, and added that as our first model of the immune response. So we had some early macrophages that would come in contact with dead cells or infected cells, begin inflammation by secreting things that would attract in T cells and others to start killing out the tumor cells. The fourth version of the model is really cool because we had another collaborator who built a lymph node compartment model. So an off-screen ODE model of basically dendritic cells and APCs leaving the simulation, arriving in the lymph node, and kind of nucleating an immune expansion, creating more cytotoxic T cells that could recognize and attack infected cells, more neutrophils, and neutralizing antibodies, and those can come back into the model. So to give you an example of the fifth version of that model, here we start with a
a lung epithelium tissue, basically, and we're going to randomly sprinkle virus particles on here. And we can see that we have this resident innate immune system that's just kind of roving and patrolling the tissue. And then, so we'll start by infecting some cells. So now that there are some cells, these yellow ones here are the ones that are infected. Some of them have been detected, particularly the dead cells, by the macrophages that are now activating and starting to kick off an immune and inflammatory response. Some of those cells are going to go off to the lymph node now. And as you see, you're going to start seeing recruitment of these new cells that are introduced into the tissue and start attacking the infection until finally, well, there's a lot of damage, but now the infection is under control and has been eliminated. So it's kind of, this nice innate and then adaptive response. We have the innate response where you kind of have some macrophages and other cells that are already resident, kind of holding the infection at bay, things like interferon responses, while the cavalry can come up and ramp up their army and come back in and finish off the infection. So here we have a fairly good model based upon that shows both the innate and the adaptive immunity components. But the interesting thing now is now you can take this immune system that we just trained by the prior infection, and now we have the neutralizing antibodies and some of the now adapted T cells already resident in the tissue and in the lymph node. And we can say, now let's kind of reset the simulation. Let's start off with a fresh patch of epithelium, say someplace that had not been previously infected, and let's do a secondary exposure. So now we simulate this, and we have a few cells that die, but the immune system is right there and ready to ramp up immediately, and the tissue damage is minimal. So you can see here where the same now model shows that the prior exposure is protective for subsequent exposure. So that's kind of a neat emergent thing with this kind of agent-based model. And the cool thing is agent-based models are very reusable. We can take the hypotheses and the code we started building for this COVID-19 model and then readapting it to cancer, where now you have cancer cells being attacked by T cells rather than infected epithelial cells. But a lot of the code and a lot of the modeling processes are the same. So we're taking these same codes and now applying them to things like digital twins. So this is kind of an introduction to what an agent-based model is, some of the key concepts and PhysiCell models that we've seen, and some examples of some fairly complex agent-based models built in the PhysiCell platform, and the way we're taking it to different conditions, particularly in cancer. Over the next several days, we'll be, in the next several sessions, we'll be further exploring how to build these models ourselves. So we want to thank a variety of funders for this work. Thank the Jane Koskinas Ted Juvalis Foundation for Health and Policy, Breast Cancer Research Foundation for earlier funding, more recent funding from the National Institutes of Health and NCI in particular, and from Leidos and the NSF. And we want to thank all of you and our community for really making this work possible and for joining us today. So we'll look forward to continuing with you in session 1 next. Thank you.